Hi Tarunika. Very good evening Tarunika. So today we are going to discuss is about cell the unit of life. Yes. Good evening Parthasarthi. Very good evening. I remember the questions. I'll send it to you very fast. Okay. Today we are going to discuss is about cell the unit of life. As you know, some 6 questions may come from this chapter. So, 6 into 4 is 24 marks we may get from this lesson. So, how to prepare? You may know, like, we have to concentrate on the numbers, like years of the discovery, scientist name. Okay. So, these things will be the task for us. Other than that, some direct questions will also come, and there won't be any tricky or questions and all. Hi Tilak, very good evening. Hi Anishri, very good evening. Okay, so we are just giving introduction about cell, the unit of life. As I said, 6 questions may come from this chapter, and of course 24 marks. And where and all you need to concentrate is about years and scientist name. All these we were discussing, Arnishri. Yes, Tarunika, very high. Fine. So let us start the class with a small funny question, okay? Can you tell me who is the sibling of E. coli bacteria? So as a cell, it is a prokaryotic we are going to discuss. So E. coli bacteria, who will be the sibling? Yes, my dear Tilak, Parthasarathi, Tarunika, and Arnishri, the sibling is of E. coli bacteria. Are you thinking? Yes, Arnishri. Your time starts. I'm counting. 5, 4, 3, 2. Any guess? Any guess, children? Tilak is thinking so sincerely. Yes, Parthasarthi. Any guess? 1. Okay. The sibling of E. coli is. I am going to reveal the answer, my dear children. It is broccoli. Yes, Tilak. So it is broccoli, the brother or sibling of E. coli. I hope you all got the answer. Anyway, now we'll start the class. Shall we proceed to the class? So, cell the unit of life is very important chapter. Okay, Dharunika. Nice one. Thank you, Tilak. Fine. So, we are going to start cell the unit of life. You know, cell is a structural and functional unit of all living organism. Either it may be a small living organism like amoeba or a gigantic whale. You find a cell, maybe unicellular or multicellular. Okay, so it may be unicellular or it may be multicellular also. As we discussed, the unicellular example is amoeba. Multicellular, as I said, whale. So any organism, it is made up of cell only. Can anyone answer me who discovered first the dead cell? So who discovered cell? Of course, it is a dead cell. Yes, Jeevan. Very good evening. Very good evening, Jeevan. Very good evening. We just started and I am asking who discovered a cell which is a dead cell. Yes, it is Robert Hooke. Okay. And the living cell was discovered by, as you know, Anton von Leeuwenhoek. Okay. So this is about living cell. Yes, Robert Hooke is the correct answer. Arini, very correct answer. Excellent. 100% correct. And it is Leeuwenhoek who discovered the living cell. Yeah, it is Anton von Leeuwenhoek. Yes, very correct answer, Parthasarathi and Jeevan, very correct. Excellent answering. So, possible question. As you know, Robert Hooke dead cell and Anton von Leeuwenhoek living cell. And Anton von Leeuwenhoek, even you know, he is the father of microbiology. Okay. Now let us see about cell theory, and you know it is Sheridan and Schwann proposed. Sheridan was working on that during 1938 and Schwann during 1939. They proposed and Sheridan is botanist, Schwann is a zoologist. Okay. What are the postulates of cell theory? So each living organism is made up of, made up of cell. And all vital reactions are carried out in cell only. All vital means it may be biochemical, metabolic reactions are carried out in cell. Okay. And of course, there was another scientist who were, yes, 18. Sorry. Yes. Very thankful. Yes, it is 1838 and 39. Thank you, Tilak. Thank you. You are very correct. It is 1838 and 39. They proposed cell theory. I wrote 19th century. Okay. Can you tell me there was a scientist who were added in the cell theory during 1855? Can anybody remember who is that scientist? Who is that scientist who were added in the cell theory? Yes. His name starts with R. Yes, Tilak. Very correct again. It is Rudolf Virchow. Is the correct answer. Rudolf Virchow, Dharini. Very good evening, Dharini. Yes. 100% correct. Yes, Tilak, Dharini and Jeevan, Arinishri. Excellent answering. It is Rudolf Virchow. And what he postulate in the cell theory is, yes, all cells are formed from pre-existing cell. So the cells are formed from pre-existing cell is given by Rudolf Virchow. Possible question is 1855. And who postulated cells are formed from pre-existing cell, that is homines, E cellula. So E is Rudolf Virchow, homines, cellula. Yes, sir. So we discussed, of course, cell theory. I hope you are clear. And this is very important. The smallest cell is mycoplasma, and the largest cell is ostrich. You can see longest is nerve cell. Okay, so you can see the nerve cell here. Bacteria size is 3 to 5. But if you see the diagram in the NCERT, they have given 1 to 2 micrometer. As both are in the NCERT, you can follow anything based on the option given in the question paper. Because both are there only in NCERT, okay. And PPLO also, if you see, they have given 0.1 in diagram and 0.3 in writing notes. Human RBC, of course, 7 micrometer. I hope you all understood. Hi, Sivaprakash. Very good evening. Very good evening, Sivaprakash. We just now started. I was explaining the controversial points. See, sometimes they may give you the match form of questions where they mention the size on one side and cells on the other side. So, if you see 0.3 also mycoplasma, 0.1 also mycoplasma. So, 0.1 to 0.3 they could have given. And of course, bacteria. It is 3 to 5 in running notes of NCERT. In diagram, it is 1 to 2. So, you should be cautious in selecting the answer. Any one might have been given. But anything is correct only as far as NCERT it is mentioned. Am I clear to you all, children? So, we are proceeding to prokaryotic cell. As you know, cells are classified into two criteria. One is prokaryotic. Another one is eukaryotic. So prokaryotic means what? Primitive. Okay. So they have primitive nucleus. Eukaryotic means what? They have true nucleus. This is the criteria we are classifying. Yes, sir. Now for prokaryotic, of course, you know, bacteria will come, mycoplasma. All these are prokaryotic organisms. As we are going to discuss about bacteria, shall I ask one more funny question about bacteria? See, bacteria like one country very much. What is that country name? Bacteria likes which country? Is the question. Yes, come on, Dharani, Harinishri, Shivaprakash, Jeevan, Tilak, Parthasaradhi. Yes, Tarunika. Any of you? Bacteria likes this country very much. Can you tell me which country it is? Yes, Sivaprakash. 5, 4, 3, 2. Shall I reveal the answer? Shall I reveal? Yes, Pa? Are you all thinking, Dharani, any answer? The bacteria likes which country? Yes, Anishri. Shall I give a clue? It begins with G. The country name begins with G. Ends with Y. No idea, Dharani. Okay, let me write. It is Germany. Yes, sir? Germany. Yes, Dharani, you got it. What you said, no idea, you answered, Dharani. Excellent, Dharani. Yes, Tilak. Jeevan, excellent. It is Germany. Very correct answer. Excellent. Super. You're rocking today. It is Germany. Yes, Dharani, Tilak and Jeevan. It is Germany. The bacteria likes the country very much anyway. So let us proceed into the prokaryotic cell bacteria. Okay. So you know what are the bacteria comes under? Prokaryotic. It may be Arachibacteria or Eubacteria. Ah, yes, you are excellent. You answered. See, I will ask so many questions to you. You wait. After giving hint. Yes. Yes, Dharani. Today is your day. You are rocking today. You answered. So, cyanobacteria, Eubacteria, Arachibacteria, PPLO, all these comes under prokaryotic cells. Okay. And the bacteria is available in 4 different forms. You know, bacteria is available everywhere. It is cosmopolitan in distribution. And you know, can you see the rod-shaped bacteria? It is bacilli. Coccus-shaped bacteria, which is spherical coccus. Comma-shaped bacteria, Vibrio. And of course, Pyrelium-shaped bacteria. So we have 4 basic shapes are there. They may be in pair also, Diplococcus, Streptococcus, all that is there. But this much is enough for us. Yes, Sanjay, very good evening. Very good evening. Yes. So we just started, Sanjay. We were discussing about prokaryotic cells. Okay. So you know bacteria, the size. Any of you remember what is the size range of bacteria? What is the size range of bacteria? Yes, Sachin. Very good evening. Happy to see your name. Yeah, very good evening, Sachin. The size range of bacteria is, any of you remember? Yes, my friend Sachin has come. Tilak. Why you didn't? I gave notification, Sanjay. You didn't get it today. I did late today. Usually we'll do early. Okay, hereafter I'll send it early. Okay. So what is the size range? You remember? Shall I show you what we discussed? As I said, 1 to 2 in diagram, 3 to 5 in running notes. So anything you can take it. Okay, the size may be 3 to 5 micrometer is the size of the bacteria. Okay. And we discussed it is found everywhere. Yes, 3 to 5 micrometer. Yes, correct answer, Jeevan. Excellent answering. Okay, it is 3 to 5 micrometer. Yeah, 1 to 2 in diagram also is given. So, based on the option they have given you, you can select the answer. Now, let us see the details. Of course, you can see a bacteria here. The outermost covering of the bacteria is called capsule. The capsule is otherwise glycocalyx. The glycocalyx is either slimy layer or it may be a rigid layer also. Okay, so this is regarding glycocalyx, and you will be learning in 12th standard portion, of course, based on the slimy and rigid. Griffith will do experiment also. Yes or no? Then, next thing, it is followed by cell wall. So, cell wall of bacteria is made up of what? A possible question. Can anybody answer me? The cell wall of bacteria is made up of what? Cell wall of bacteria is made up of what is the question? Who will answer me? Yes, Sanjay, Jeevan, Tilak, Sachin, Dharini. The cell wall of bacteria, yes, my dear. Parthasarthi. Starts with P. Is anybody? Starts with P. I gave hint also, Dharni. The cell wall of bacteria. Yes, Arnishri, excellent answering. 100% correct. It is peptidoglycan. Peptidoglycan is made up of phospholipid, but it is peptidoglycan. Okay. So it will be having peptidoglycan, a possible question. Yes. Fine. So cell wall, this is a possible question. Yes, Sanjay, very correct answer. Peptidoglycans. So we finished cell wall. We finished capsule. Then third one, plasma membrane. So you can see the plasma membrane here. And these plasma membrane, you can find the foldings. The foldings of the plasma membrane is called what? The foldings of plasma membrane is called mesosome. Can anybody summarize or write out? Yes, Sachin, very correct answer. Excellent. It is peptidoglycan. Very correct answer. 100% you are correct. Okay, can you tell me what are the functions of mesosomes? What are the functions of mesosomes? What are the functions of mesosome is the question. Can anybody recollect? Yes, Parthasarthi, Sachin, Sanjay, Harini, Tilak, Dharini. Very good. Respiration. So question will come like this. If mitochondria is absent here, who will do the mitochondrial function? It is mesosome. Possible question. Apart from that, DNA synthesis, distribution to daughter cells. So all these are performed by mesosomes only. Am I clear to you, children? Understood? So we discussed cell wall, capsule and plasma membrane. So plasma membrane, the folding is called mesosome. Yes, might be aerobic or anaerobic. Bacteria we can't say, they are, facultative organisms. Some are parasite. Okay. The enfoldings are called mesosomes. Yes. Very correct, Arnishri and Sanjay. Excellent. Okay. The next one we are going to discuss is about the genetic material of bacteria. So, which is DNA, of course. Is it double stranded circular DNA or single strand circular DNA? What it is? Is it double stranded or single stranded circular DNA the bacteria will be having? Yes, Jeevan. Okay. The bacteria will be having double stranded circular DNA is the correct answer. Okay. Another possible question, what is the genetic material of DNA or genetic material of bacteria is called? Yeah, double stranded circular is the correct answer. Yes, select. And Arinishri, excellent answering. It is double stranded circular DNA. Fine. Can you tell me? Yes, Partha Sardi. So, can you tell me what is the other name of genetic material? Other name of genetic material is nucleoid. Dharni. So, it is called nucleoid or genophore. Next possible question. So, what is the other name of genetic material of bacteria? It is nucleoid or genophore. Okay. Then the extra chromosomal circular DNA only we call it as plasmid. The extra chromosomal circular DNA we call it as plasmid. Yes, Jeevan, very correct. Tilak, the extra chromosomal DNA is plasmid is the correct answer. Okay. And you know, plasmid acts as a vector in biotechnology also. It acts as vector. Okay. So I hope you are clear with the genetic material. Now we'll proceed to the cytoplasm. Can tell me what is the cell organelle we find in the bacterial cytoplasm? Yes, it is genophore. Excellent answer. It is genophore. Rinishri, excellent answer. 100% you are correct. Genophore, even otherwise called nucleoid. Okay. Can you name what is the cell organelle? We find it in the cytoplasm of the bacteria. Yes, Dharani, Parthasarthi, Jeevan, Sanjay. Yes, Sachin. What are the cell organelle? Can you name it? Name the cell organelle. What do we find in the cytoplasm of bacteria? Yes. Yes, Jeevan. Excellent, Jeevan. You are very correct. 100% correct answer, Jeevan. And Parthasarthi, it is ribosome. Yes, ribosome we find. The only cell organelle we find is ribosome. Yes, Tilak, it is ribosome. No doubt it is ribosome. Excellent answer. ER and all we are not having, my dear Sanjay. Ribosome we are having. Ribosome is the membrane bound cell organelle. True or false? Ribosome is the membrane bound cell organelle. True or false? Is the membrane bound cell organelle. Do we have membranes on ribosome? Good evening, Subhan. I'm thinking of you. Very good evening. Or watching match, my dear. Yes, false is the correct answer. We just started, Subhan. We were discussing about bacteria. And ribosome is the non-membrane bound cell organelle. Okay. Is it 70S or 80S we found in the ribosome? Is it 70S or 80S we found in the ribosome? Yes, Sachin. We do not have membrane. Yes, Jeevan, correct answer. And is it 70S or 80S? We found in the ribosome. The ribosomes are 70S or 80S. Yes, Sanjay, very correct. It is 70S. Is the correct answer. So we have 70S ribosome. Yes, Arnishri and yes, Dharani, that was a false statement and Parthasarthi 70S ribosome is the correct answer. Okay. And you know, ribosomes are the protein factory. So they involve in protein synthesis. When many ribosomes aligned involved in protein synthesis, it is called what? Next possible question. Many ribosomes aligned like a chain involved in protein synthesis is called what? This structure. Yes, it is polyribosome. Again, Sanjay, very correct. Excellent answer. It is polyribosome or polysome. Yes, Sanjay Tilak, very correct answer. Yes, it is polyribosome or polysome. That is the only cell organelle. We can find it in the bacteria. You can see the ribosome here. Yes, Jeevan and Parthasarthi, it is polysome. Very correct answer. Excellent. Fine. Then apart from that, we find inclusion bodies. So inclusion bodies like a vacuole you can assume. Bacteria does not have vacuole. Instead, they have inclusion body where they reserve synophysian granule, glycogen granule. Okay. And also gas vacuole, a storage space like vacuoles they are, no? We have even gas vacuoles are found. Can you anybody tell me gas vacuoles are found in which are the bacteria? We find gas vacuoles. Yes, Subhan. Gas vacuoles are found where? Is the question. Gas vacuoles are found in is that me? Blue-green bacteria, purple-green bacteria. Yes, cyanobacteria, very correct answer. Blue-green bacteria and purple-green bacteria and also photosynthetic bacteria. Yes, very correct answer. Tilak and Sanjay again excellent answering. 100% very correct. It is blue-green. Okay. So blue-green and then purple-green. Photosynthetic is the correct answer. Yes, Sachin. Very correct answer. Yes, Subhan, you got the answer now. Gas vacuoles. Okay. Then flagellum. So flagellum is used for, yes, Nishri, it is blue-green algae. Bacteria, cyanobacteria is the correct answer. Okay. Yes, photosynthetic. Yes, Subhan. Flagellum, of course, used for locomotion. So it is used for locomotion, okay. And you can find many finger-like projection. You are going to identify the answer now. Finger-like projection. Many finger-like projection helpful to attach on the harder surface is, yes, locomotion is correct. Yes, flagella is for locomotion. Very correct answer. 100% that is correct. That is over. Now my next question is, they are finger-like projection helpful to attach on the harder surface is what? Is it pili or cilia? Cilia. Do they have cilia and flagella on the same? Yes. It is Sai Asutosh. Your villain for NEET. Anyway, it is fimbriae is the correct answer. Yes, fimbriae is the correct answer. Excellent answering, Sai. Very good evening, Sai and Tilak. You are also very correct. Excellent answering. It is fimbriae. Yes, Anishri. Fimbriae is the correct answer. 100% you are correct. Okay. And pili are these projections. They form conjugation canal. Yes, Jeevan fimbriae is the correct answer. So I hope you are clear with the pili also, fimbriae also, flagellum also. Yes, sir? So this is regarding the structure of bacteria. Now let me ask, yes, I. Now I am going to ask you one funny question. Let me see whether you are answering for this question. Why bacteria are poor in maths? Why bacteria are poor in maths is the funny question. Yes, I. Why we say bacteria are bad in maths? Yes, my dear Sanjay, Jeevan, yes, I, Tilak, and Dr. Chowdhury. Very good evening. I am noticing your name now only. Subhan, Sachin. Any answer? Why bacteria are poor in maths? Ah, yes. You both combined the answer. Actually, yes, they multiply by dividing. Okay, that is the answer. So they multiply. One will become two when they undergo fission. So they multiply by dividing is the correct answer. Yes. Yes, Tilak, you answered correctly. Excellent. Yes, Sanjay. Yes, you are 100% correct, Sanjay and Sachin and Subhan. Tilak. Excellent answering. So you answered and you are laughing. Very correct. Okay, I hope you understood the prokaryotic cell and the possible questions. Cell wall is made up of water. Yes, Jeevan, it is multiplying by dividing is the correct answer. Okay, and as you know, mesosomes, the next possible questions here. Okay, what is the genetic material of bacteria? Many ribosome aligning, locomotion by flagella, finger-like projection fimbriae, conjugation canal pili, or these are possible questions. So, this is regarding prokaryotic cell. I hope you are all clear with the prokaryotic cell theory. We are proceeding to eukaryotic cells. So, you know, eukaryotic cell, they have plant cell, animal cell, and fungal cell. All these, you know, even protista. All these are eukaryotic only. So, we are going to discuss the components of the eukaryotic cell. The first one we are going to discuss is plasma membrane. After 1940s, after the invention of electron microscopy only, we could able to visualize the plasma membrane. What is plasma membrane? It is ultra thin, elastic, semi-permeable, non-living, limiting, membrane. One of the word is wrong here. Can anybody guess which word is wrong here? Yes, Sanjay. Yes, Sai. Harini. Tilak. Chaudhary. One word is wrong here. Which word is wrong? Is it ultra thin wrong or elastic or semi-permeable or non-living or limiting? Which word is wrong? Okay. Actually, Arini is correct. It is living membrane. So, non-living is the wrong statement. Very correct. Excellent. Subhan and Parthasarathi. Yes, I. And Jeevan. Yes. Excellent answering. It is a living, limiting membrane is 100% correct answer. Okay. And you know, plasma membrane. The first discovered plasma membrane is from RBC, which is called red cell ghost. Yeah, it is semi-permeable only. Selectively it permits. Means semi-permeable only. No, half of the things will be permeable. Half of the things it may not allow. That is the meaning, na Sai? So it is red cell ghost. RBC first plasma membrane, which we call it as red cell ghost, and you know the composition. So what is maximum in plasma membrane? Is it lipid or it is protein or it is carbohydrate? Which will be maximum? Yes, I. Which will be maximum in plasma membrane? Is it lipid or protein or carbohydrates? Yes, Sanjay. Subhan. Yes, Sai. Excellent again. It is lipid. Very correct answer, Sai. Excellent. 100% you are correct. And Tilak, Parthasarathi, 100% correct. Yes. Jeevan, Harini Shri. Very correct answering. Excellent. So the composition, we have lipid is maximum and the two it is phospholipid. Then protein will be there. Of course, later on only you have carbohydrate. Hardly 5% is carbohydrate, 79% is lipid. But if you take RBC, you find 52% is protein, 40% is lipid. It's an exception. So in general, yes, my dear Mercy, we were wondering for the funny questions and all answers. Now you were coming late. Yes, very good evening, Mercy, my dear. Okay, so we have 52% protein and 40% lipid in RBC. Okay, and if you see the structure, as you know, the well accepted model is fluid mosaic model. Can anybody tell me who proposed this fluid mosaic model and which year? Yes, I. Dr. Chaudhary. Yes, Sanjay. Hi, Mercy. Sanjay is telling. Who proposed the fluid mosaic model, pa? Anybody? Oh, you slept. No, it's not Crick and Watson, my dear Dharmi. Singer and Nicholson is the correct answer. Yes, Sai, you're rocking today. Yes, super. It is Singer and Nicholson in 1972. Yes, Sanjay also correct. Yes, Jeevan also correct and Tilak correct. It is 1972 and Singer and Nicholson proposed the model of fluid mosaic model. Okay, so I always ask you to assume like you were standing in front of Antarctica Ocean. Yes, Arnishri and yes, Mercy. Tilak, yes, it is Singer and Nicholson, 100% correct answer. Yes, Martha Saradi and Subhan. Like we are all standing in front of Antarctica Ocean. Okay, what we could able to visualize there? Of course, a beach means wave will be there always. Above the wave, there will be floating icebergs also there. Yes or no? So the waves are called lipids. So in a lipid sea, proteins are floating. This is what they said. Proteins are floating. In fluid mosaic model, of course, you can see the lipid bilayer will be like this. You have hydrophilic head, hydrophobic, non-polar tail. Okay. Now let me give you one statement. Let me see who is answering correctly. Lipids flip-flop movement. Shows the flip-flop movement. Okay. Whereas proteins cannot. Is the correct statement or wrong statement? True or false? The lipid flip-flop protein cannot. Is it true or false? I'm waiting for your answer. 5, 4, 3, 2. Okay. Actually, it is true only. Okay, true is the correct answer. Very correct answer, Aranishri and Tilak, Dharini, Parthasarthi and Jeevan. Excellent answering. 100% correct. I'll tell you what is flip-flop movement. Flip-flop movement is vertical movement. Okay. So it vertically move from one corner to another corner means flip-flop. So lipids can move flip-flop. You can imagine the wave na? From one end to another end it is coming wave. Whereas protein icebergs are not moving vertically. It may move left side to right side like that. Horizontally, there is no synchronized movement for the icebergs. So, lipids only flip-flop, whereas protein cannot. However, it will show movement, but it is not a flip-flop movement or vertical movement. I hope you are clear, children. Okay, so this is another possible question in terms of statement type questions. Okay, and you should know which is hydrophilic and polar head hydrophilic, non-polar tail is hydrophobic. This is another possible statement type questions. Okay. Now, what are the functions they perform? Of course, can anybody tell me what are the functions it performs? Yes, Jeevan, Subhan. Why we need a membrane? What is the function of membrane? Yes, it acts as a pump. Diffusion. Yes, very correct answer. 100% correct. Sanjay and Arinishri, excellent answering. Any other? Yes, it involves itself in transport. Phagocytosis, very good. You're rocking today, Arinishri. Excellent answering. Phagocytosis it perform. Yes, Jeevan, diffusion is the correct answer. Yes, I. Osmosis, very correct answer, I. Excellent answer. Okay, so transport it perform. So what is the transport? It may be diffusion or it may be active transport or it may be osmosis even. Yes, Jeevan, osmosis even. Okay, or so these are all, they are involved with the cell membrane and phagocytosis, cell eating. Which are the cell involved in phagocytosis? There are two blood cells will involve in phagocytosis. Can anybody answer me? Yes, Arini Shri. Rocking today. Pinocytosis also performed by plasma membrane is very correct answer. Excellent answering. Yes. Pinocytosis means cell drinking. So it takes the liquid molecule. And immunocytosis, cell vomiting. Yes, excellent. You're rocking. WBC cells will do phagocytosis, especially neutrophil and monocytes. Very correct answer, Sai. Excellent, Sai. Yes, it is neutrophil and monocyte will do. Yes, monocyte and neutrophil. Monocyte and neutrophil is the correct answer, Jeevan. And Sai was rocking today. Yes, excellent. Yes, Jeevan. So these are the functions of cell membrane. Okay. I hope you are clear with plasma membrane. Yes, it is neutrophil and monocyte. Very correct answer, Arnishri. Any doubt on cell membrane? Anybody has any doubt? Shall I proceed to the cell wall? Yes, Subhan. Parthasarthi. Any doubt? Sanjay. Okay. Actually, cell wall is for protection. The cell wall is found, no doubts. Okay. So cell wall, you can find it in the plant cell. Of course, in fungi. Yes, thank you, Subhan. Thank you, Jeevan. No doubts. Okay. And also in the some of the protists. Okay, not all protists. Some protists will be having. Of course, amoeba and all may not be having. So cell wall is present. It is not found in animal cell as you know. So cell wall, we have primary cell wall and secondary cell wall. The secondary cell wall lie inner to the primary or outer to the primary. The secondary cell wall lie inner to the primary or outer to the periprimary. Anybody? Yes, Tilak, you're rocking. It is inner to the primary. So secondary cell wall is found inner to the primary is the correct answer. Subhan, Arini, Sanjay and Jeevan, very correct answer. So it lies inner. In between primary cell wall and secondary cell wall, we find middle lamellae. Okay, so this middle lamellae is made up of calcium pectate and magnesium pectate. Okay. And middle lamellae of one cell wall connected to middle lamellae of another cell wall is called what? Another cell wall is called dash. Anybody? See, middle lamellae of one cell wall connected to middle lamellae of another cell wall. This only I am asking. What is it called? Any guess? Starts with P only. Very correct. Excellent, Arini Shri and Dharini. Yes, it is plasmodesmata. Yes, plasmodesmata is the correct answer. Sai, excellent Sai and Tilak. Yes, it is plasmodesmata. Very correct answer. Excellent. Okay. Yes, Jeevan. Plasmodesmata is the correct answer. I hope you all remember. Simplest transport need plasmodesmata. Plasmodesmata will be more in dividing cells. These are also possible questions. Okay. Now, what are the other things? See, you find some pores also in the pores or pits in the cell wall. Again to transport. Which cell wall has meristematic activity? Is it primary cell wall or secondary cell wall? Has meristematic dividing capacity. Meristematic activity found in which cell wall? Is it primary or secondary? Yes, very correct. Arinishri again, excellent. It is primary, which actually divides because it is having parenchyma cells. Yes, Tilak, it is primary, which has dividing capacity. Yes, Parthasarathi. And Jeevan, very correct answer. Excellent. It is primary, which has the meristematic activity keeping on divide, which is primary cell wall. Okay. And the cell wall composition is cellulose in case of plants. Cellulose, hemicellulose and pectin. Of course, in case of fungi, it is chitin. In case of protista, it may be, silicaceous cell wall. They are having cellulose is there in case of dinoflagellates. So varies. Fine. What are the functions of cell wall? What are the functions of cell wall? What are the functions of cell wall? Yes, very correct, Dharini. Yes, what is that emoji? I didn't understand, my dear Sanjay. Yes, it gives shape, rigidity, protection is very correct. What is it? So, functions, it gives rigidity. Oh, protection. Okay, rigidity. Yes, protection. Yes, Dharini, transport. Yes, I. Protection and preventing from bursting. Very correct answer, Sai. Excellent. Yes, Jeevan. Structural protection and gives shape. Yes, very correct answer. So, protection, transportation also it will do. And prevent bursting, of course, is the correct answer. So, all these are the functions of prevent from bursting, maintaining the shape. So you have various shapes in cell, na? So all that is maintained because of the cell wall only. I hope you are clear with the cell wall. Understood, children? So today let me tell you a small topic which is related to practice. As you know, practice makes man Perfect. So Subhan likes cricket. You know your Thala Dhoni practice for how many hours? Okay, what are emoji and all you are putting? I didn't understand, Sanjay. What does it mean for? We were in Globe. So how many hours our Thala Dhoni practicing? Any idea? Thala Dhoni ki he clapped. Subhan. He practiced for 4 hours every day. You know, he is champion of champion. Even then he practiced. Okay, 4 hours. 2 hours he will do batting. 1 hour he will do fielding. And 1 hour, of course, he is also doing bowling. So 4 hours every day he is allotting for practicing. Oh, 8 hours, huh? That is Saturn. Okay. Fine. So practice is needed even for you people. Okay. A small story also I would like to share with this. உங்க எல்லாருக்கும் ஒரு சின்ன கதை நம்ம எப்பவுமே சொல்ற மாதிரி ஒரு சின்ன கதை வருது. இந்த கதை என்ன சொல்லுதுன்னா ஒரு பையன் இருக்கான். அவனை வந்து அந்த காலம் நாட் அவர் கரண்ட் ஸ்டேஜ். ஓகே. அப்ப என்ன பண்ணுவாங்க, குருகுலத்துல கொண்டு விடுவாங்க. அப்ப குரு கிட்ட அவன் போய் படிக்கிறான். ஓகேயா. அவன் படிக்கும் போது அவன் பேர் வரதராஜன். அவன் படிக்கும் போது அவனுக்கு படிப்பே வரல. அவன் எல்லாரு கூடயும் நல்ல வேலை செய்யறான், குருவுக்கு நல்ல வேலை செய்வான், அவங்க ஃப்ரெண்ட்ஸுக்கெல்லாம் நல்ல ஹெல்ப்ஃபுல்லா இருக்கிறான், எல்லா வேலையும் கிளீன் பர்ஃபெக்டா பண்ணுவான். ஆனா படிப்பு மட்டும் வரவே வராது. அவன் கூட படிச்சவங்க எல்லாம் வந்து oh you are very much worried. Even Virat Kohli practicing for 4 hours தான். சரியா? Don't worry, be smile. Okay. So விராட் கோலியுமே வந்து 4 மணி நேரம் டெய்லி பிராக்டிஸ் பண்றாரு. அண்ட் அத பிராக்டிஸ தவிர்த்து அவர் வந்து எக்ஸசைஸும் பண்றாரு. கார்டியாக் எக்ஸசைஸ் எல்லாம் பண்ணிக்கிறாரு. அதுதான் அவரோட லீன்னஸ் அண்ட் வெரி ஸ்டாமினா அண்ட் மசில்ஸ்க்கான காரணம் வந்து என்னன்னா அவரோட அந்த கார்டியாக் எக்ஸசைஸ் தான். ஸோ அவரும் பண்றாரு. நான் யாருக்கும் அகேன்ஸ்ட் எல்லாம் இல்லை. எஸ் சுபான், கப்பு கொடுத்துட்டா எனக்கு சுபான். இந்த கதையோட கண்டினியூஷனுக்கு போவோமா? கதை என்ன சொல்லுதுன்னா அவன் ரொம்ப ஹெல்ப்ஃபுல்லா இருக்கான். ஒன்லி திங் அவனுக்கு படிப்பு மட்டும் வரல. சோ அவன் கூட படிச்சவங்க எல்லாம் அடுத்த லெவல் போயிட்டாங்க. எல்லாரும் குரு குலத்தையே விட்டு போறாங்க. ஆனா இவன் மட்டும் அடுத்தடுத்த பேட்சுக்கு வந்து who is BK I don't know. Oh PK our PK. Okay, fine. He is my friend. What to do? ஓகே. சோ அப்ப என்ன பண்ணுவான், இவன் மட்டும் குருகுலத்திலேயே இருப்பான். ஒரு ஒரு ஸ்டேஜுக்கு மேல குரு கூப்பிட்டு சொல்லுவாரு, கண்ணா உனக்கு படிப்பு வரல, உங்க அப்பாவும் கஷ்டப்பட்டு பணம் அனுப்புறாரு, நீ வீட்டுக்கு போயிட்டு அப்பாவுக்கு உதவியா இரு அப்படின்னு சொல்லிடுறாரு. அப்ப இவன் ரிட்டர்ன் போறான். ஊருக்கு போகும்போது நிறைய காட்டு வழியா போறான். எஸ் மர்சி, ஐ காட் இட். எனிவே எஸ் திலக், ஐ காட் இட். அப்ப போகும்போது ஒரு கிணறு பாக்குறான். ஓகே. அந்த கிணறுல வந்து ஒரு ராக்னால கட்டியிருக்காங்க ஆக்சுவலி. அதுல பாத்தீங்கன்னா அந்த கோடா இருக்கு. அப்ப அவன் கேக்குறான், அந்த கோடு வந்து எப்படி வந்தது அப்படின்னு கேட்டா, அந்த கயிறு போட்டு இழுக்கும் போது வர்ற கோடு தான். ஓ, இந்த கயிறுனால அந்த ராக்லயே வந்து கோடு விழுந்திருக்கா அப்படின்னு சொல்லி அவனுக்கு ஒரு தெளிவு வருது. திரும்பி வேகமா குருகுலத்துக்கே போயிடுறான் ரிட்டன். அப்போ குரு பாக்குறாரு, இல்ல நான் படிப்பேன் அப்படின்னு சொல்லி சொல்றான். அவனோட எந்தூசியாசத்தை பார்த்து குருவும் அவனை சேர்த்துக்கிறாரு. அதுக்கப்புறம் அவன் எப்படி படிச்சான்றதுதான் அவனோட ஸ்ட்ராட்டஜி. அவன் படிக்கும் போதெல்லாம் என்ன பண்ணுவான், ஒரு கப்புல நெல்லு கொண்டு வந்து வச்சுப்பான். வச்சுட்டு ஒரு வாட்டி படிக்கிறதையும் ஒரு நெல் எடுத்து வச்சுவான். திரும்பி அதையே படிப்பான், இன்னொரு வாட்டி நெல் எடுத்து வைப்பான். திரும்பி படிப்பான் நெல். ஒரு ஸ்லோகம் ஆர் வாட் எவர் இட் இஸ், அதை வந்து அவ்வளவு டைம்ஸ் அவன் ரிப்பீட் பண்ணியிருக்கான். அதுக்காக அவன் கவலைப்படல. சோ ரிப்படிஷனா நிறைய வாட்டி படிச்சதுனாலதான் அவன் வந்து குருகுலத்துல எத்தனையோ பேர் படிச்சு ஒரே வருஷத்துல பாஸ் ஆனவங்க எல்லாம் நம்ம பேசவே இல்லை. ஏன் இந்த வரதராஜனை பத்தி பேசுறோம் அப்படின்னு பாத்தீங்கன்னா, அந்த வரதராஜன் தான் பின்னாடி சமஸ்கிருதத்துக்கே இலக்கணம் எழுதினார். ஓகேயா. சோ வந்து பிராக்டிஸ் ரொம்ப முக்கியம் மக்களே. அதுக்காக தான் சொல்றேன். நீங்க எல்லாரும் நல்லா படிக்கிறீங்க. படிக்கிறீங்க படிப்பீங்க எல்லாம் எனக்கு தெரியும். ஆனா கொஸ்டின் பிராக்டிசிங் இஸ் வெரி இம்பார்ட்டன்ட். அவர் எப்படி 2 மணி நேரம் பேட்டிங்கு, 1 மணி நேரம் ஃபீல்டிங்கு, 1 மணி நேரம் போலிங்னு வச்சுக்கிறாரோ, அதே மாதிரி நீங்களும் 2 மணி நேரம் படிச்சீங்கன்னா கண்டிப்பா 1 மணி நேரம் நீங்க வந்து பிராக்டிஸ் பண்ணணும். அண்ட் விராட் கோலி என்ன செய்வாரோ, அவரோட பழைய வீடியோ எல்லாம் பாப்பாராம். நம்ம எங்க தப்பு பண்ணிருக்கோம் அப்படின்னு அவரோட வீடியோவே அவர் பார்ப்பாரு. அதை வந்து அவர் ரெக்டிஃபை பண்ணுவாரு அடுத்த மேட்ச்ல. அதேதான் நம்மளும். நம்ம கொஸ்டின் பேப்பர் வாங்கிட்டோம், எழுதிட்டோம், மார்க் வந்துச்சு, தூக்கி போடக்கூடாது. நம்ம எங்க தப்பா பண்ணோன்றத கண்டிப்பா பார்க்கணும். அண்ட் மத்த 3 ஆன்சருக்கு என்ன மாதிரி கொஸ்டின்ஸ் வரும் அப்படின்றதும் ரொம்ப முக்கியம். சரியா? டிப் டு ரிவைஸ் how to do for each chapter. ஒரு சாப்டருக்குமே இதுதான் கான்செப்ட். முதல்ல அண்ட் ஃபோர்மோஸ்ட் திங் நான் எப்பவுமே சொல்றது அதான், நம்மளோட பைபிள், குரான் ஆஸ் வெல் அஸ் பகவத்கீதா எதுன்னா நம்மளோட என்சிஆர்டி பயாலஜி புக்கு தான். என்சிஆர்டி பயாலஜி புக்கை நீங்க தரோவா படிச்சுடுங்க. அது கூட சேர்த்து கொஞ்சம் எக்ஸ்ட்ரா பாயிண்ட்ஸ் அப்படின்றது இந்த கிளாஸ் நோட்ஸ் தான். கிளாஸ் நோட்ஸை நீங்க புக்லயே எழுதினாலும் சரி, இல்ல ஒரு நோட் போட்டு தனியா அந்த டாபிக் வச்சிருந்தாலும் சரி, இல்ல பிட் பேப்பர்ல எழுதி புக்ல ஒட்டிண்டாலும் சரி, எதுனாலும் ஓகே. உங்களுக்கு எது கன்வீனியன்ட்டோ அண்ட் படிக்கும் போதே நீங்க ஹைலைட் பண்ற பழக்கத்தை கொண்டு வாங்க. ஹைலைட் பண்ணீங்கன்னா இப்ப படிக்கும் போது ஃபர்ஸ்ட் டைம் ஒரு சாப்டர் படிக்கும் போது 6 மணி நேரம் குறைஞ்சது எடுக்கும் இல்லையா? அப்ப நீங்க மெதுவா படிச்சு ஹைலைட் பண்ணிட்டீங்கன்னா எக்ஸாம்க்கு முன்னாடி நீங்க ஹைலைட் பண்ண பாயிண்ட்ஸை மட்டும் பார்க்கலாம். ஒரு சிலரோட புக்கெல்லாம் பாத்தீங்கன்னா பூரா புக்கையுமே ஹைலைட் பண்ணிடுறீங்க. அப்ப எது ஹைலைட்டடு? அட்லீஸ்ட் வேற வேற கலர் யூஸ் பண்ணீங்கன்னா கொஞ்சம் டிஃபரன்ஷியேட் பண்ணி பார்க்கறதுக்கு யூஸ்ஃபுல்லா இருக்கும். அது பண்ணலாம். சரியா? இந்த இயர்ஸ், இந்த சயின்டிஸ்ட் நேம் இந்த சாப்டர் பொறுத்த வரைக்கும் இப்ப படிச்சு முடிச்சதுமே இப்ப சில டாபிக் எல்லாம் நீங்க எழுதியும் பார்க்க வேண்டியிருக்கும். ஃபார் எக்ஸாம்பிள் நம்மளோட போட்டோசிந்தசிஸோட சைக்கிள்ஸ், ரெஸ்பிரேஷன் சைக்கிள்ஸ் எல்லாம் எழுதி பார்த்தாதான் நமக்கு புரியும். அந்த ஏடிபி பேலன்ஸ் எல்லாம். சோ அந்த மாதிரி சிலது எழுதி பார்க்குற மாதிரியும் இருக்கும். டேபிள் ஃபார்ம்ல எழுதிக்கலாம், மைண்ட் மேப்பிங் பண்ணிக்கலாம், ஏதோ ஒண்ணு பண்ணிடலாம். எது பண்ணாலும் நீங்க அன்னைக்கு என்ன டாபிக் படிச்சீங்களோ அந்த டாபிக் வந்து ப்ராக்டிஸ் பண்றது ரொம்ப மஸ்ட். மினிமம் 200 question a day you should practice. Yes, sir. Yes, sir. You are CBI not 7. நீ கண்டுபிடிச்சிட்டு. ஓகே. எஸ். ஸோ வந்து நீங்க என்ன பண்ணணும்னா இதுதான் வந்து டிப்ஸ் ஃபார் ஸ்டடிங் ஈச் சாப்டர். சிம்பிளா சொன்னா என்சிஆர்டியை படிங்க, கூட கொஞ்சம் கிளாஸ் நோட்ஸும் பாருங்க, ப்ராக்டிஸ் பண்ணுங்க. ப்ராக்டிஸா வந்து நெவர் ஸ்டாப் பண்ணாதீங்க. நான் அதுக்கு தான் தலை தோனி இங்க இழுத்து வந்தேன். தலை தோனி வந்து இன்னைக்கு நேத்து கிரிக்கெட் விளையாட பல காலமா விளையாடிட்டு இருக்காரு. அவர் எவ்வளவு கிரவுண்ட்ஸ் பார்த்துட்டாரு, எவ்வளவு மேட்ச் பார்த்துட்டாரு. ஆனாலும் அவர் எவ்ரிடே 4 hours ப்ராக்டிஸ் பண்றாரு. அப்படின்னா என்ன அர்த்தம்? நம்மளும் நிறைய நேரம் ப்ராக்டிஸும் பண்ணணும். அப்பதான் நமக்கு ஹெல்ப் பண்ணும், மார்க் வாங்க. என்ன மை டியர் மர்சி, திலக், சுபான், சஞ்சய் அண்ட் பார்த்த சாரதி, am I correct or not? எஸ்ஸா? இன்னைக்கு கதை உங்களுக்கு கொஞ்சம் ஃபயர் வந்திருக்கும்னு நினைக்கிறேன். இந்த ஃபயரோட போய் கொஞ்சம் படிச்சு என்சிஆர்டியை தரவா கொஞ்சம் ரிவைஸ் பண்ணி ப்ராக்டிஸும் பண்ணுங்க. ஓகேயா? ஸோ மீதியை வந்து நம்ம நெக்ஸ்ட் கிளாஸ்ல வந்து we can discuss endomembrane system and other than endomembrane system, of course. So any doubts, please stay back and ask me. If you like the class, ஒரு லைக்கும் போடுங்க. Thank you all. Yes, Subhan, thank you for your fire. Yes, Tilak. Yes, Sanjay, thank you. Thank you, Jeevan. Thank you, Mercy. Thank you, Subhan. Bye, Mercy. Thank you, Tilak. Bye. Thank you, Arni. Good night. Thank you, Jeevan. Thank you, Parthasarthi. Thank you, Arni. You rocked it today. Keep rocking. Bye. What doubt, Sanjay? Yes, tell me, what is in TCA cycle? Yes or no? How to find carbon products? What carbon products TCA cycle? See, முதல்ல உனக்கு TCA cycle என்ன வரும்? அசிட்டைல் கோஏ இல்லையா? இந்த அசிட்டைல் கோஏ வந்து OAA ஓட சேரும். இந்த OAA ல எத்தனை கார்பன் இருக்கு? 4 கார்பன். அசிட்டைல் கோஏல எத்தனை கார்பன் இருக்கு? 2 கார்பன். இது ரெண்டும் சேர்ந்ததுன்னா உங்களுக்கு என்ன வரும்? ஆக்சுவலி citric acid, which is 6 carbon. என்சைம் உங்களுக்கு தெரியும்னு நினைக்கிறேன். ட்ரைகார்பாக்சிலிக் அண்ட் டைகார்பாக்சிலிக். ட்ரைகார்பாக்சிலிக் வந்து உங்களுக்கு ஆக்சலோ அசிட்டிக்கும் மேலிக் ஆசிடும் தான். சரியா? டைகார்பாக்சிலிக் ஆசிட் வந்து ஆல்பா கீட்டோ குளுட்டாரிக் ஆசிட். 2 கார்பாக்சைல் குரூப் இருந்ததுன்னா அது டைகார்பாக்சிலிக். 3 கார்பாக்சைல் குரூப் இருந்ததுன்னா ட்ரைகார்பாக்சிலிக். சோ அந்த அளவுக்கு ஸ்ட்ரக்சரல் டீடைல்ஸ் நமக்கு என்சிஆர்டிலயும் கொடுக்கல. So you need not worry that much, I think so, Sanjay. But இதுதான் ஆன்சர் ஆக்சுவலி. டைகார்பாக்சிலிக் அப்படின்னா 2 கார்பாக்சைல் இருக்க குரூப் வந்து பாத்தீங்கன்னா நம்மளோடதுல ஆல்பா கீட்டா குளுட்டாரிக் ஆசிட். 3 கார்பாக்சைல் குரூப் இருக்கிறது சக்சினிக் ஆசிட். சரியா? Am I clarified your doubts? Yes. Okay, shall we finish off the class? Thank you, Sanjay. தெரியலையே. Might be. Yes, sir. Any other doubt? Thank you. Bye. Bye, Tilak. Bye, Sanjay.